So hello everyone, welcome to another particular lecture series, and this particular lecture series we are talking about asymmetric synthesis, which is the advanced part of a stereochemistry course. So this particular asymmetric synthesis are not that much important for the students who are preparing for JAM examination. However, some question was there in the GATE and CSIR NET examination, and this particular chapter is very important for those students who are in the final year of undergrad or final year of post graduation, because normally institute offers asymmetric synthesis as a course. So for those students this chapters is very important. JAM people might skip, but GATE and CSNet people, you can watch this particular lecture. However, this particular asymmetric synthesis is very important in the industrial purpose and all, and mostly the research work has been now currently shifted to this type of synthesis. Now, before entering that particular asymmetric synthesis, let's understand what is the asymmetric synthesis is. Like asymmetric synthesis, it is a reaction in which a achiral unit in an ensemble of substrate molecule is converted into a chiral unit in such a manner that unequal amount of stereoisomer are produced. For example, suppose you were thinking about a prochiral center, and after doing some kind of substitution reaction or some kind of addition reaction, a chirality is generated. So that type of synthesis is not that much called the asymmetric synthesis. Like suppose you are converting a prochiral compound to a chiral compound. Now there is a two type of stereoisomer can be formed. Like it can be formed either a R enantiomer or can be formed the S enantiomer. Now the asymmetric synthesis is termed when you are converting a prochiral compound to a chiral compound, but maintaining one enantiomer is major over other. For example, in kind of SN1 reaction, the initial stage the carbocation has been formed, and that particular carbocation is a planar one. Now that particular nucleophile can either approach from the bottom end as well as it can approach from the above end. So there is a uh, both the enantiomer has been formed, and we call this racemic mixture. So this is not a asymmetric synthesis. If somehow that one enantiomer will be major one, then we can call it asymmetric synthesis. And this type of asymmetric synthesis had greater importance in the industrial purpose, as such as the drug discovery, drug development, as well as the biological system. Also the asymmetric synthesis is very important, because asymmetric synthesis enables the selective formation of one enantiomer over another, which is crucial in drug development because our body is chiral, which actually stimulate with certain enantiomer more over other enantiomer. For example, if you have seen my stereochemistry lecture, in lecture 1 I have talked about the thalidomide disease. Now this type of thalidomide having two different isomer, R isomer and S isomer. Now that particular R isomer is actually the drug which actually address the morning sickness problem for the pregnant woman, whereas the S isomer is not the drug. Actually it cause harm, it causes genetic damage to the embryo. So in the previous time, when the industry people and the doctors are not that much aware of this type of chirality, asymmetric synthesis, the racemic mixture, stereoisomer type of thing, they produce the both isomers, because the chemical formula are the same for that case, just the orientation different. So that's why when that type of thalidomide drug has been given or the prescribed to the pregnant woman, then what happened, that R isomer will do that particular job, but since there is a S isomer is also there, that will also cause genetic damage to the embryo. So when this type of report has been frequently reported in the medicinal field, then people are actually, the scientists, the researcher are actually looking onto it, that what is happening in that particular things, and that the asymmetric the importance of one enantiomer in the human body is actually discovered, and from that onwards the asymmetric synthesis comes into the picture. Because when you have to prepare a thalidomide drug in the industry scale, now the industry scale only focus to form the R isomer, because S isomer is no longer to be used in the drug. So they have to find certain protocol where this R isomer, this I isomer would be the major one. So from there actually asymmetric synthesis has been observed. And as well as if we dig down in our human body itself, we'll found that our human body is also asymmetric, where our body contains the enzyme or kind of ribosome, and all kind of things are actually followed in the certain one chirality over another chirality. This is, you can call the natural selection or something, anything. Even if you look about the nature itself, nature is also asymmetric. They are also leaning towards, more biased towards one enantiomer over other enantiomer. For example, in our body, the proteins that are actually generated from amino acid, in our body natural selection, the protein are built exclusively from the L enantiomer of amino acid. Except glycine, glycine is the prochiral amino acid, otherwise the 19 essential amino acid all are L enantiomers. And this is because that the present of the enzyme and the ribosome present in our body, which are chiral and which are very very stereospecific, where only the L amino acids are actually naturally selected by our enzyme and ribosomes, and that's why our body actually recognize and incorporate L amino acid over D amino acids. So if we feed the body D amino acid, our body's enzyme and ribosome are not detected at all. So it generally won't be used properly, or maybe metabolized differently, or even be toxic in nature, because our body is designed to incorporate L amino acid. Now in future, if any kind of foreign or alien body has been discovered by scientists which can consume D amino acid, the enzyme and ribosome present in that particular foreign or alien body which are actually recognize the D amino acid, then there will be utility of this D. But for our cases, in our body scenario, the L amino acids are mostly recognized and incorporated. And very interestingly, whether you can see the nature or in our body, our the reactions schemes are very very efficient, that only the L amino acids are actually formed. D amino acid is never formed, like 100% one stereoisomer has been formed over other enantiomers. So our body, the nature are very very efficient to form this type of asymmetric synthesis, where in laboratory scale it might not that we reach the 100% enantiometric excess till now. Okay, now if we think that if we look about how nature produced the corresponding asymmetric synthesis, or the asymmetric enantiomer major over the other one, we as well as the scientists have been found that those particular nature as well as our body which contains chiral compounds, which chiral compounds actually influence to form one stereoisomer over another stereoisomer. Like in our body, as I already discussed, that enzyme and ribosome are present, they are actually the chiral one. They actually preferred one enantiomer over another enantiomer. So from there scientists elucidate that maybe a chiral influence are actually required to form asymmetric synthesis. So from there this question has been come, that how a simple reaction where you are using a prochiral compound and you make it chiral, however you want to do a asymmetric route where one stereoisomer would be major, so how can we do that particular thing, that how a simple reaction can be turned into asymmetric one? So from the nature they have discovered that a simple achiral reaction become asymmetric when you introduce a chiral influence. That means a chiral influence is necessary to form a particular asymmetric synthesis. Now, how can it do? It can be a chiral catalyst has been introduced in the particular reaction mixture, which actually biased uh, to form in one stereoisomer over another, or it can add a chiral auxiliary, a chiral reagent, or substrate control. Now, this particular substrate control has been observed in our body itself. Like in our body, you can find that the reaction vessels are kind of protein pockets in our body, and that protein pockets have certain kind of chirality, certain kind of orientation, which actually hinders the corresponding substrate to come from one side. So they restrict the path of the substrate coming into the protein pocket from one side, which actually leads to formation of this type of one stereoisomer major. That means the asymmetric root. So this type of chiral influence can be affect the substrate by either implying steric hindrance that is observed in our human body itself, where the protein present in our human body are actually limits the entry point of the substrate by implying the steric bulk of this particular protein, globular protein, or you can say any the proteins, helical chains and all. Or another thing is that when the chiral catalyst can be used, they can generate a intermolecular hydrogen bonding during the transition state, which actually uh, influence to form one enantiomer over another enantiomer, or the ligand assistant. Like the ligand have the dual role, either the ligand will be chiral as well as the ligand can also produce steric bulk. So this type of pathway, uh, can the chiral influence has been observed, and this particular thing influence to generate one stereoisomer major over the other stereoisomer. Now let's summarize all this thing with a simple SN1 reaction. Now let us summarize all the point by using this part of SN1 reaction. Here you can see that this particular center, chiral center. Okay, now after during the SN1 reaction, this is the intermediate. This is the sp2 carbon right now. This is a carbocation has been formed now, and this particular carbocation is a planar one. Now that particular nucleophile has two options. It can come from the bottom end, or it can come from the above end. So if there is a no that type of influence is there, then there is a probability to come from bottom end and the above end is the same. So that's why we got the 50% of one stereoisomer, 50% of another stereoisomer, and this is called the racemic mixture. And if we see the transition state of this particular reaction, then we can found that this is the intermediate, which undergo either nucleophile come from the bottom end or nucleophile come from the above end. Both the cases you can find that particular the energy is the same, and that's why the two different product formation rate is equal. That this particular stereoisomer can be formed in 50%, this particular stereoisomer can form in the 50%. This is the same energy gap, or that means the equal amount of product. Now if we add a chiral influence here, maybe that particular chiral influence can be do some kind of steric bulk to one of the side. For example, we can say that if we somehow hindered one side attack, for example, if you attack something like this, the chiral influence hindered here. This is a chiral influence. So the nucleophile approaches from this side having the lower energy barrier Ts over nucleophile approaching from this side have the higher energy barrier, because a chiral influence is there which actually restrict the nucleophile to attack from the backhand side. So the activation energy of that particular case is increased manifold over the nucleophile comes from the non-hindered side. So naturally the reaction follows this particular pathway more, which result to form this particular product as a major one, and this minor. Now if that particular chiral influence is so strong that no nucleophile can attack from the backhand side, then we can say that the major product might have nearly 99% EE. That means 99.5% of this product and 0.5% of this product. Maybe it may be the enantiometric axis is 100%, but mostly 99.5% has been observed in the laboratory scale frequently. Okay, so uh, in a simple way, we can now say that unequal ratio of product observed and one of the stereoisomer is prominent over the other one, and welcome, you are entered into the asymmetric synthesis. And thus the asymmetric synthesis has been designed as well as the asymmetric synthesis has been done in the laboratory scale as well as the industrial scale. So with this we have to end this particular lecture. In the next lecture we are talking about some stereochemical models such as the Cram model, Cram-Chillet model, Falkin-Allen model, and that particular model part is useful for the IIT JAM, CSI, NET and GATE examination. So do watch that particular lecture, and this particular lecture I hope you learn the gist about the asymmetric synthesis. So we'll meet on the next lecture. So thank you for listening.